Good morning, dear students. Our lecture for today is about humoral immunity. Now, in order to understanding humoral immunity, first you need to take a short break remembering these outlines, just like what are the components cellular component of innate immunity. The cellular component of innate immunity include neutrophil, eosinophil, basophil, mast cells, macrophage, monocyte, dendritic cell, and natural killer cell, NK cell. What about the cellular component of acquired immunity? Just like T and B cell, T and B lymphocytes. T lymphocyte, it's the main player of cellular immunity, and B lymphocytes are the humoral immune response main players by antibody production. T cell, as you know, T cell include T helper, T cytotoxic, T regulatory T cells. Each one provide, such as T helper cell, provide assistance to other cells. T cytotoxic T cell killing viral infected and tumor cell, and T regulatory regulation of immune responses. Now, why do we need humoral immunity in fact, while we already have innate immunity? Although innate responses are important in protection against pathogen, they are not fine-tuned to particular antigen and do not generally improve upon repeated encounter with the same infectious agents. Now, other questions. Let us ask this question. Explain why innate immunity does not improve upon repeated encounter, as I said before, with the same infectious agent. Why? Because it's not specific for the antigen, and there's no immunological memory. While in acquired or adaptive immune response, it's specific to the antigen, and there will be immunological memory. And also let us discuss something about the antigen. To go deep through humoral immunity, you have to understand the role of antigen in electing immune responses. You already knew from previous lecture that there are specific features for antigen as immunogen. You know, not all antigen are immunogen. And there are many factors. The immunogenicity of antigen depends on the dose and the structure of antigen as hapten or complete antigen, the site of administration, and other factors. We are not going through these factors because you already knew from the first lecture. And also, you have to know the role of immune cell in activation, assisting B cells. Why B cells? Because B cells are the main player of humoral immunity. These cells, these lymphocytes, produce antibodies against the infectious agents or its antigens or its toxins. Okay. Now we mentioned that acquired immunity is specific for particular antigen. Let us stop here in that point. What is the antigen-specific recognition molecules in humoral immunity, for example? Of course, the recognition molecules in humoral immunity are antibodies. Now, you have to know the structure of antibody, just like the antibody have two Fab. We mean by Fab the antigen binding site, and there's variable regions and constant region. And in constant region there is a FC region. This FC region bind with phagocytic cell, NK cell, with complement proteins. Also, you have to know the classes and subclasses of antibody immunoglobulin, and also you have to know why we call these immunoglobulins. What's the reason for that? And the classes, each class, and function of each class, just like IgG, IgM, IgD, IgA, and IgE. Also, the subclasses of IgG, for example, IgG1, GG2, GG3, and GG4. Also, you have to know the function of antibodies. The functions of antibody, like opsonine, we mean by opsonine leading to phagocytosis and activation of complement classical pathway, antigen recognition through IgM and IgD on B cells. Now, what is the antigen-specific recognition molecule on cellular immunity? I mean on T cells. Now, T cell cannot recognize and bind with the whole antigen molecule as in the case of B cell. T cell can recognize and bind with antigenic peptide after processing of antigen by antigen-presenting cell. You know what's antigen-presenting cell? Antigen-presenting cell that present antigen to other immune cells, just like phagocytic cell, dendritic cell, and many other cells regarding as antigen-presenting cell. Okay. In that case, when T cell bind with the processed peptide presented by antigen-presenting cell, it should be complexed with MHC class 1 or 2 regarding to the type of antigen, of course. If antigen is bacterial antigen or bacterial cell, bacterial antigen, bacterial toxin, in that case, the antigen-presenting cell will complex the peptide, processed peptide, with MHC class 2, and then this complex will be recognized by service marker on T cell called CD4 T cell. It's CD4 plus T cell. That means that this T cell have service marker called CD4. When binding through these two receptors, the CD4 service marker and the complex of antigen MHC class 2, then there will be a second signal to activate the T cell through binding with another set of receptors between the antigen presenting cell and the T lymphocyte. Also, if it's viral infected cell or tumor infected cell, in that case, CD8 T cell surface marker will bind with MHC class 1 complex with the processed peptide from the viral infected cell or tumor infected cell, and then other combination of two binding receptors on the antigen presenting cell and T cell in order to elect the activation of T cell in that case. We mentioned that antibody has a role in activation of classical pathway. We are going the classical pathway of complement. We will go deep through this subject in complement system lecture. Okay. Now, antibody can activate phagocytosis through opsonization. That means antibody can act as opsonin. As we know, microorganisms are sometimes able to resist phagocytosis, but if small amount of antibody are added, the phagocyte springs into action. That means it will be activated. Through recognition of two or more antibody molecules bound to the microbe using specified Fc receptors. As you remember in this previous slide that we mentioned in antibody structure, there are in the constant region, there are Fc regions which will bind to many cells such as NK cell, phagocytic cell, and also it will bind with complement proteins. In that case, the phagocytic cell have specific receptor to Fc region called Fc receptor on the cell surface of the phagocyte, and this will bind with Fc region of antibody that opsonize the antigen or pathogen. We are going to explain factors affecting the clearance of pathogens by opsonins, mediated phagocytosis, and antigen-antibody interaction lecture. These factors include valency, such as univalency or multivalency. These factors affect the phagocytosis. Now, what are other activities of antibodies? There are many activities in addition to phagocytosis activation or complement activation. There are many activities such as directly neutralize viruses, prevent bacterial adhesion to body surfaces, neutralize bacterial toxins, antibody-dependent cellular cytotoxicity, formation of immune complexes, to enable removal of antigen from circulation. In that case, the triggering of mast cell, eosinophil, basophil, degranulation, and this point we will discuss it in details in hypersensitivity lecture. Also, it's very important to have an idea about T and B lymphocyte because both can interact during humoral immunity. As I mentioned before, that T helper T cell will assist B cell in antibody production. Also, very important item called clonal selection. You have to understand what is the clonal selection. We mean by clonal selection that the antigen select lymphocytes that possess the specific receptor for this antigen. Each B cell is programmed to make only one specificity of antibody, and it have membrane receptors on its surface, just like IgD and IgM. These membrane transmembrane immunoglobulin, the IgD and IgM, it's just like receptors for antigen. These act as receptor for specific antigen. In fact, each cell have 10 to the power 5 antibody molecules on its surface, all of identical antigen specificity on its surface. B cell will be specialized to become plasma cell in that case, and which produce large amount of soluble antibody in their endoplasmic reticulum when B cell will be activated in order to produce antibody. Then antibody is secreted from these plasma cells into circulation or into the local environment to encounter the antigen which elect these antibody production. Now, when antigen enters the body, it will be only bind to receptor on B cell that fit with its epitopes. Why this happen in that case? Because as we mentioned, acquired immunity, including the humoral and cellular immunity, is specific and have memory. That means the antigen will be remembered from these cells. B cell, after binding with antigen, then develop into plasma cell and memory B cell. Okay, as we mentioned here. In that case, as B cells are programmed to make specific antibody which can recognize the same antigen that previously bounded to the surface immunoglobulin on B cell. In this way, antigen selects for the production of antibodies that recognize that. Here is the mean of specificity. Each B cell will produce antibodies specific to the antigen which elect the immune response or the antibody production. What about T cell? This is the same way. In the same way, T cells will, with a TCR receptor, TCR, T cell receptors of appropriate specificity are selected for the antigen. These cells are required to help B cells proliferation and differentiation into plasma cell. In this figure, antibody-mediated activation of phagocytosis explained by this figure. Here, as you see, the binding of a microbe to a phagocyte by multiple antibodies give strong association forces and triggers phagocytosis. By by making a cross-linking the surface by Fc receptors on phagocytic cell with Fc region on antibody. Now, the immunological, we mentioned immunological memory. Immunological memory and how it is important in subsequent exposure to the same antigen. You can find details about primary and secondary immune responses in immune response lecture of undergraduate students. Okay. Here I have an idea about different types of T and B cells. How T helper cell play a role in B cell activation, and as we already knew from previous lectures that different types of lymphocytes are all derived from common lymphoid progenitors. Hematopoietic stem cells develop from either myeloid or lymphoid pathways of cellular differentiation. All lymphocytes develop from lymphoid roots, and thus the common lymphoid progenitor produce both T and B cells. The T lymphocyte possess either gamma delta or alpha beta T cell receptors. Those with alpha beta T cell receptor and alpha beta T cell, that's mean T helper 1, 2, regulatory T cell, Reg, T reg, suppressor T cell, cytotoxic T cell. The major divisions of B cells is B1 and B2 population. It's the most important. There are many divisions of B cells, B5 or other B cells, but the main are B1 and B2. You have to know the difference between these subdivisions. Now, something important you have to know, and we will discuss this. And you can find more details in undergraduate student lecture. I mentioned the primary response and secondary response. Now the primary and secondary immune response. During the first encounter with the pathogen, with its antigenic determinant, for example, bacterial antigen, this will elect primary immune response. This response characterized by slow onset because naive lymphocytes need time to proliferate into sufficient numbers. Here, the primary immune response then will fade quickly. While in the second encounter with the same antigen, the immune response, the secondary immune response is rapid and more intensive. The reason behind this is the generating of memory cells during the primary immune response. That's the memory B cell. The generation of memory cells provide the basis for vaccination in that case, where the immune response is primed by relatively harmless forms of microbial antigens so that immune system directly generates secondary immune response upon the first encounter with the actual pathogen. In a primary immune response, IgM first generates, then followed by IgG. In secondary immune response, the intensity of IgM stayed the same as in primary response, while IgG levels increases in that case and stay prolonged period of time, providing prolonged protection against pathogen. Also here, you have to know more details about the antibody, their structure as we mentioned, classes, function of each classes in that case. Also, it's recommended for you to have an idea about membrane receptors. For antigen, as in B cell serves receptor for antigen, B cell receptors. We mentioned something about that in previous slides, such as IgD, IgM. These are membrane service receptor in B cell, on B cell service. It's a membrane receptor for antigen in that case. Also, the generation of diversity for antigen recognition. You have to know this information, okay? Thank you very much.